Hello and welcome in Vienna, Austria. We're here at TU Wien and my name is Weiss. Today I want to talk to you in this video about gas phase electrophoresis on our NES GEMMA instrumentation and its application in bio-nanoparticle research. The topic of this video will be gas phase electrophoresis, applying an NES GEMMA instrumentation as a highly versatile analytical technique in bio-nanoparticle or nanoparticle characterization. And to start with this video, I want to give you a short overview on the topics which we will discover in the next 25 minutes. First, I want to introduce you to our instrumentation, which is the NES GEMMA instrument, before going into detail what we can learn generally from our NES GEMMA data, and then I will go into detail concerning results in virus and virus-like particle research, as well as research concerning liposomes and extracellular vesicles. In the end of the video, I want to give you a short conclusion and maybe outlook to what will be done in the near future. So what about the instrument? You can see it here on that slide. That's our GEMMA instrumentation. GEMMA is short for gas phase electrophoretic mobility molecular analyzer. As you can see here, that instrument is also known under several other names like macro IMS, LiquidScan ES, ES-DMA, or ES-SMPS. That makes literature research sometimes a bit hard, but all of those instruments are doing the same thing. They're separating single-charged analytes in the gas phase at ambient pressure according to their electrophoretic mobility diameter. And in case of spherical analytes, that electrophoretic mobility diameter corresponds to the size of the particles or the size of the analytes. And even when we talk about proteins, for instance, these will collapse in the gas phase and also act as spheres. And in the case we have some structures which are not spherical, we will have some pseudo-spherical analytes. The instrument itself consists of three parts. More to that in a minute, but the first part you can see here, that's the so-called nano-electrospray aerosol generator with a charge reduction device. Secondly, we have the electrostatic classifier with a nano-differential mobility analyzer. The nano-differential mobility analyzer, or DMA in short, that's that silver tubing on the side. That is where the magic takes place. And last but not least, we have an ultrafine condensation particle counter as detector unit, as you can see here on the right-hand side marked in green. For the first part of our instrument, the nano-electrospray source, we are applying our analytes in a volatile electrolyte solution. Analytes are then electrosprayed by application of pressure, by application of an electric field and a sheath flow at the tip of a cone-tipped capillary. We are generating droplets, and these droplets are drying down, and at the same time, charge equilibration occurs. Charge equilibration will happen in a bipolar atmosphere induced, for instance, by a 210 polonium alpha particle source, a soft X-ray charger, a corona discharge process, or many other possibilities. We end up with mostly neutral particles, which we will not discuss any further. A certain percentage of analytes is single charged, and in these particles we are interested in. These particles we then take to the second part of our instrument, which is the nano-differential mobility analyzer. There, two forces are acting on our analytes. First, we have a tunable electric field, and in an orthogonal direction, we have a high sheath flow of ambient air. By variation of the field strength, it's possible to size separate the particles which we have electrosprayed previously, and by this, it's possible to get a monodisperse aerosol or monodisperse aerosol fractions out of the polydisperse aerosol which we have generated in the nano-electrospray source of the instrument. The monodisperse or monomobile aerosol fractions we are then taking to the detector unit of our instrument, which is the condensation particle counter. There, we have a supersaturated atmosphere, and as particles enter that atmosphere, droplets will again grow, and they will be counted as they pass a focused laser beam. And that's our instrument in action. You can see here on the one hand side a sample chamber where we can put our sample to the nanoelectrospray source. You can see the capillary leading the sample then to the spray chamber, where on the right hand side we have our corona discharge process. Particles are then transferred to the nano differential mobility analyzer, which can be seen on the side of the instrumentation. And last but not least, we have our condensation particle counter, counting our particles after separation. What we then get is a spectrum, as you can see here, for an exemplary protein. On the x-axis, we have the electrophoretic mobility diameter. On the y-axis, we really have the particle counts, as it is possible to count particles in the detector unit. This is in accordance with the recommendation of the European Commission for nanoparticle characterization, as we are number-based. This allows us to detect monomeric species next to dimers, trimers, or even multimers of proteins, and it allows us to detect smaller particles next to larger ones. Now, what can we learn from such data? In principle, it's possible to learn data concerning the size of the analytes, sample purity, stability of our particles. We can learn about the aggregational behavior, or we can deduce the molecular weight of our analytes. Alternatively, we can use our NES GEMMA instrumentation also as size filter by keeping the voltage constant in the DMA unit. By this, only particles of a certain size are able to pass through the size filter, and these particles can then be collected on sample supports for subsequent analysis, applying orthogonal methods like microscopic techniques, mass spectrometry, spectroscopy, or dot plot analysis. This has been shown for quite a number of analytes like proteins or oligonucleotides or carbohydrates, viruses, virus-like particles, and so on and so forth. I tried to give you a list of all those analytes which have been analyzed so far, and to sort them at least slightly according to the particle size. With this video, I now want to concentrate only on several of those analytes. To be precise, I want to concentrate on viruses and virus-like particles, liposomes, and extracellular vesicles as analytes. Now, what about virus-like particles? These days, I think everyone is knowing what a virus is doing and how a virus looks like. To keep things easy, a virus normally is a proteinaceous shell. Sometimes you also have a lipid envelope, and inside such a virus particle, you have genomic information. Virus-like particles are deduced from such virus particles. However, they do not contain any viral genomic material, which means it's quite safe to work with them. They can be applied for vaccinations, for instance, or in gene therapy as cargo carriers, as it is possible to encapsulate genomic information other than the viral genome inside the VLPs. Now, what can we learn for VLPs or for viruses in general when applying gas phase electrophoresis on our NES GEMMA instrumentation? On this slide, you can see that it is possible to deduce the size of VLPs from gamma spectra. On the left-hand side, you can see our gamma results for a VLP based on an adeno-associated virus-like particle. The numbers which we get here for the size of those VLPs are in good accordance with numbers which we get, for instance, from atomic force microscopy. Some of those results you can see here on the right-hand side of that slide. We can also check for the purity of our preparations. This time we were using human rhinovirus. I'm sure every one of us knows such a virus. That's the virus causing common cold infections. And what we did here is we took such a preparation to gas phase electrophoresis and found that such a preparation not just contains virus particles, but also some contaminating material, which was of approximately the same size as virus particles. In the end, it took us some years to figure out how to get rid of this additional contamination. But in the end, we succeeded and ended up with highly pure virus preparation batches of a human rhinovirus. It's also possible to check for the stability of virus-like particles. Some examples you can see here. On the left-hand side, you can see results for norovirus-based virus-like particles. Luckily, these were just VLPs and not the real stuff of viruses. But as you can see, for low size values, for low EMD values, we get signals for proteins which are forming those virus-like particles next to virus-like particles which can be seen at approximately 35 nanometers EMD. Another example would be SARS-CoV-2. Again, here we analyzed virus-like particles and saw also some building blocks in the lower EMD range. And these building blocks were changing over time, indicating that the virus-like particles in solution were not stable, but started to dissociate over time. It is also possible to check for aggregation of such analytes, again, showing some results for adeno-associated virus-like particles. On the left-hand side, you can see GEMMA and AFM results for a preparation or for a fraction just containing monomeric species. On the right-hand side, in contrast, you'll see a fraction which also contains some oligomers. You can see those as well for gamma spectra and also for AFM for microscopic data. And last but not least, it's also possible to derive the molecular weight of an analyte in question from gas phase electrophoresis data. The starting point for that was in 2001 by Gerold Bacher and colleagues, who found out that by analyzing proteins of different molecular weight, they got different electrophoretic mobility diameters. Thus, it was possible to set up a correlation between the electrophoretic mobility diameter and the molecular weight, this time for proteins. Now we asked ourselves, is it also possible to get such a correlation for virus-like particles? Indeed it was, and we analyzed several virus-like particle preparations and different virus-like particle species, and also had some molecular weight values which we got from literature. And by combining those data, it was possible for us also to set up a correlation between the electrophoretic mobility diameter of such species and their molecular weight. Now, what can we do with such an information? Based on such a correlation, it is possible to calculate the molecular weight of a VLP in question based on its electrophoretic mobility diameter. This we have shown, for instance, with human papillomavirus 16 or HPV 16, which was not possible at the time where we did those measurements to be measured with native mass spectrometry. However, as you can see here, it was possible to get a nice spectrum from our gas phase electrophoresis data, and by applying our correlation between the electrophoretic mobility diameter and the molecular weight of VLPs, it was possible for us to calculate the molecular weight of such a species. That molecular weight was in excellent accordance with the expected molecular weight based on the building blocks of such analytes. Another example would be adeno-associated virus-like particles. Again, it was not possible to deduce the molecular weight of such analytes from native mass spectrometry. We got a signal, as you can see here, but we were not able to use charge deconvolution to get down to the molecular weight value. However, as I've shown you, we got fairly nice gas phase electrophoresis data, and by applying our correlation, it was possible for us to calculate the molecular weight of such a species. Again, that value was in excellent accordance with data which we got from literature, as well as data which we in the end also got from native mass spectrometry. So to conclude, for virus-like particles, it's perfectly possible to get size data, purity data, stability data, and data concerning aggregational behavior and the molecular weight of virus-like particles. And we can use our NES gamma instrumentation as a size filter for subsequent microscopic detection or also dot plot analysis. These data I have not shown you today, but it's also possible to check for the immunogenicity of such particles later on after size collection. And now to something completely different. The second type of analytes which I want to present you in this video are actually lipid vesicles. For us, starting point for our investigation were actually liposomes. What are liposomes? When you have lipids, these molecules have a hydrophobic and a hydrophilic part. When you take them to an aqueous medium, an aqueous buffer, you will get all kinds of aggregates, micelles or liposomes. Liposomes consist of a lipid bilayer and have an aqueous interior. And in these particles we took interest in. Actually, we prepared those liposomes ourselves, as you can see here on the right-hand side of that slide. We used a dried lipid film, which we then hydrated with ammonium acetate solutions in order to get to our liposome samples. Now the question is, what can we learn about those lipid vesicle containing samples by applying gas phase electrophoresis? One thing which we can learn concerns the size of the liposomes. You can see here some of our initial results when we analyzed our liposome sample with gas phase electrophoresis. We get really nice signals at approximately 80 nanometers EM diameter, and no matter what sample dilution we're using, the peak remains more or less at the same position. This indicates that we do not have any unspecific aggregation, plus it was possible for us to size collect vesicles from such a sample on a solid sample support and do AFM measurements later on. Again, some of these results you can see here on that slide. I have to stress that with gas phase electrophoresis on our gamma instrumentation, we get the surface dry particle size in contrast to, for instance, DLS measurements, where we get hydrodynamic particle diameters. And this you can see here as well on that slide. There is a difference between surface dry liposome diameters and hydrodynamic particle diameters. Plus, what is also of interest, as we have a number-based particle detection, with gas phase electrophoresis, we are capable of detecting also smaller sized sample components inside a liposome sample. In that case, these are analyzed at approximately 20 nanometers EM diameter, and these might correspond, for instance, to micelles or unspecific lipid aggregates. So besides information concerning size and purity of preparations, it's also possible to check for the stability of liposome samples. We checked for ultrasonication of samples, and indeed were able to follow how ultrasonication disrupts lipid vesicles. You can see that here on the left-hand side. Also, it was possible for us to check for the repeatability of analyte preparation, and for that lipid composition which we were using at that time, we got some really nicely repeatable and reproducible results. Additionally, it was possible to use our gamma instrumentation as a size filter for the analysis of liposomal samples. What did we do here? We size collected liposomes of a certain size on a solid sample support, and then wanted to use multi-mass spectrometry for their characterization. In order to do so, we used a multi-matrix, we applied it to the sample support and applied the whole thing to a sample carrier in order to introduce it to our mass spectrometer. And we were not just interested in liposomes per se, but also we used VLDL as an additional component containing lipids. Some of the results you can see here on the right-hand side of that slide. On top, these are multi-mass spectrometry results for our liposomal containing sample. We get peaks for the lipids which we used for preparation of our vesicles. Below, you can see the results for VLDL. Again, some signals corresponding to lipids present in such particles. These signals, however, are of course different than for our lipids in liposomes. When mixing those sample components together, we get a mixed spectrum, as you can see on the third trace. That trace consists of lipids found in liposomes as well as in VLDL particles. And what did we do then? As I've said, we size collected liposomes at a size where we were sure just to get liposomes on our sample support, and then we took such a sample to our mass spectrometer. And what we found there were just signals corresponding to lipids which we were using in our liposome preparation, which means by using NES-GEMMA or gas phase electrophoresis as a size filter, it's possible to discriminate two lipid-containing species inside a sample. But that was not the only thing what we did. I've shown you already some microscopic data concerning particles which we size collected with our GEMMA instrumentation. Of course, we also did that with liposomes, and some of the results you can see here. We size collected liposomes. Some of them contained cargo molecules, others didn't, and results concerning AFM were comparable. But it was also possible for us to do spectroscopy for individual liposomal carriers. And on that slide, you can see the results. In red, we obtained spectroscopic data for cargo molecules which were encapsulated inside a single liposomal vesicle. In green, you'll see the spectroscopic data for the lipids which form the liposomal vesicles. And then below in trace F, you can see the overlay of both signals. To conclude, with gas phase electrophoresis and by application of our GEMMA instrumentation as a size filter, it's possible to size collect individual cargo carrying liposomes on a sample support, and then doing spectroscopic investigations of single individual vesicles. So to sum up, with gas phase electrophoresis, it's possible to get data on liposomal size, on purity of preparations, stability of vesicles, as well as aggregation. And it is possible to use such an instrument as a size filter for subsequent microscopic analysis, mass spectrometric or spectroscopic characterization of particles. However, we were not just interested in liposomes, but also in their native counterparts. Those native counterparts are so-called extracellular vesicles. These are vesicles also containing of a lipid bilayer, which are shed by cells and are used, for instance, for cell-cell communication. We prepared such extracellular vesicles from raw milk and took our sample then to gas phase electrophoresis. As you can see here on the left-hand side, indeed, we found some signals corresponding to EVs. Also, it was possible for us to size collect those particles for subsequent AFM analysis. It was not just extracellular vesicles from milk which we analyzed, but we also took interest in EVs derived from serum. And we obtained such samples and checked the content, the EV content of such preparations, applying several techniques. Of course, gas phase electrophoresis on our NES-GEMMA instrument, but also mass spectrometry we used, and last but not least, also nanoparticle tracking analysis, or NTA. That technique enabled us to follow the vesicle content in samples during sample preparation. And some of the results you can see here, we did some buffer exchange prior to gas phase electrophoresis, and NTA enabled us to follow the size and the concentration of particles inside such a sample. Taking such a sample then to gas phase electrophoresis, we saw not just signals in the higher EM diameter range, but also some signals at approximately 5 to 10 nanometers EMD. We concluded that these signals correspond to proteins, and these proteins are either co-purified with extracellular vesicles during the purification steps, or they are sticking to the surface of extracellular vesicles and are thus vesicle attached. Of course, we also took interest in those proteins, and it was possible to do an STS page gel to excise the bands and do mass spectrometric identification, at least for the most abundant proteins which we found there. It was possible for us to identify some of the proteins, and the results were in excellent accordance with the peaks which we found when doing gas phase electrophoresis. We then tried to purify EV-containing samples further. And for doing that, we focused on size exclusion chromatography. And again, some of the results I can show you here. When doing size exclusion chromatography, we lost some of the particles, as you can see here based on NTA results. However, the general size of vesicles seemingly stayed constant. However, when we did gas phase electrophoresis on our GEMMA instrumentation, we found that all for a sudden vesicles were less stable, as you can see here on the right-hand side with the blue trace and those spikes which you can detect there. These spikes indicate that the vesicles are no longer stable, and we concluded that the proteins which were found in the original sample had the purpose of keeping the extracellular vesicles intact. As soon as you remove those proteins from your sample, the vesicles will no longer be stable and will start to fall apart upon gas phase electrophoresis, at least in part. So to conclude that part, it was possible for us to check with gas phase electrophoresis on our GEMMA instrumentation for the size of extracellular vesicles, for the purity of preparations, as well as for the stability of such analytes. And we could use gas phase electrophoresis as a size filter in order to collect EVs for subsequent microscopic characterization of particles. That brings me now almost to the end of that video. I hope I could show you that NES Gamma yields much information on bio-nanoparticles. I also checked some of the literature, and I checked with PubMed. And since the start of the method in the late 1990s until now, I found approximately 217 publications. Why approximately? Because not all of them are actually listed in PubMed. But let's see, 200, 250 publications are out there. And from those 200 to 250 publications, approximately 50% have a connection with Vienna University of Technology or TU Wien. I also hope I could convince you that gas phase electrophoresis offers some really nice possibilities for on and offline hyphenation to orthogonal analytical techniques. And what we will do in the future is we'll focus on bio-nanoparticles in special biological matrices. So please stay tuned for another video, and please also check the other videos which you can find on the net. In the end, my thanks go, of course, to you for watching this video, as well as for some of the funding agencies, and of course to the team. And special thanks to Günther and Ernst, and they passed sadly away in 2022, but they are in our memories. Thank you very much for joining. Thanks for joining me for this video. Please also check the other videos which you find from my side on YouTube, and see you very soon again in Vienna. Bye!